Welcome to the lecture series on vitamins and nutrition. Today, as a continuation from the previous session, we are going to look at fat soluble vitamins and water soluble vitamins, their fundamental difference, and then we are going to proceed further into the study of water soluble vitamins. So, there are many water soluble vitamins. So far we have done a full round of fat soluble vitamins. So, it is high time I thought like I can refresh little bit about the difference between water soluble vitamins and fat soluble vitamins. So, as students who study the basics, especially specializing in, I would say, nutritive sciences, you need to be well aware of the advantages and disadvantages in the food which are rich in water soluble and fat soluble vitamins, and how we basically take this into consideration when we really process foods. See, first particular point, what we consider as to be the use or the role that water soluble vitamins and fat soluble vitamins play in our normal diet as part of a nutrition. So, water soluble vitamins, they act like precursors for coenzymes and also they act as antioxidants, whereas fat soluble vitamins also act like coenzymes. One more step ahead, they act like hormones. It means what? They are produced from one particular part of the body, in this case liver being a principal site, where they are produced, synthesized and being released, okay, like vitamin D for that matter, you could consider it to be a kind of an hormone. Okay. So, they regulate variety of metabolic functions, and yeah, obviously they also act like antioxidants. We have seen the role of vitamin E and we have seen the role of vitamin K playing a role, but in this aspect functionally both water soluble and fat soluble vitamins look alike, but if you closely monitor, water soluble vitamins act like precursors for coenzymes. In other words, there is a coenzyme which has been metabolically important in the normal I would say physiology of our body, like the way we utilize carbohydrates, the way how we utilize proteins and in all the anabolic and catabolic functions. So, we have coenzymes. So, you need water soluble vitamins to make these coenzymes, that is why it is called precursor. So, as such if you say whether they directly go and act like kind of a coenzyme, not necessarily. So, they are the raw material for the raw material. I would say, I will not say raw material, you could say the raw material for the coenzymes, and without this precursor the essential coenzymes may not be produced, whereas fat soluble vitamins are little different over here because they are full fledged coenzymes, although there is this part of a precursor role being played, but we can't exactly call it precursor because these fat soluble vitamins are initially in the inactive form and then they get converted to active form, whereas if you use this word precursor, it means they are used as one of the raw material so that they would assemble and form the final coenzyme. So, that way the final coenzyme being formed would require fat soluble and water soluble vitamins, but more predominantly the water soluble vitamin, whereas fat soluble vitamin would directly act like a coenzyme. And as I have told you just now, as they are also kind of an hormone. As far as the antioxidant role is concerned, like scavenging free radicals, yes, both have a equal role to play in their own way and the way kind of mechanisms or the metabolism that they contribute to. Okay, so, that way they are similar, but in this aspect they are little different. Now, second thing, suppose if you end up taking more amount of water soluble vitamin, so what will be the status? So, if you are going to take more amount of water soluble vitamin, it may not be very toxic. That is something very interesting to note. Even you would have noticed this whenever you take excess of any vitamin rich in water soluble components, water soluble vitamins, you would definitely notice that there would be a change in the color of the body fluids, right from the color of the saliva and right from the color of the urine output. you would see it would be a little more concentrated. This is an indication that this excess of water soluble vitamins being eliminated through kidneys are being released from our body. So, there is very little chance that they would stay inside your body, but that is not the case for fat soluble vitamins because they are hydrophobic in nature fundamentally. So, they have a very high propensity to stay in our tissues, especially in the adipose tissue, in the liver. So, that is the fourth point. That is the third point you need to notice. So, they are not stored except vitamin B12, whereas most of the fat soluble vitamins are stored and especially stored in the liver, probably kidneys or maybe those fat rich adipose, but predominantly liver being a primary organ for storage and synthesis of fat soluble vitamins. So, since they are stored, our body can run without these vitamins for a little longer period of time. So, you do not need to like regularly take it. That is one of the reason why even identifying deficiency for fat soluble vitamins takes a while, because we still depend on the dietary source, I mean depend on the stored reserves present in our body, and we can go along for a long period of time, and there would be a point where we would not even realize that we have depleted all the resources, then the symptoms of deficiency starts surfacing. So, probably if a person is going to make any dietary changes, and maybe the diet what they are trying to experiment are probably available to them, if it is being deprived of a fat soluble vitamin, they may not see the difference. So, they may think it is okay with the diet and could proceed for a longer period of time. Eventually they will start showing symptoms, but that is not the case for water soluble vitamins. So, how our body would cope up with that? So, we have to have regular, I would say, supplementation of water soluble vitamins, and moreover this water soluble vitamins, even a single, I would say, lack for a period of 3 to 4 days, okay, eating a diet deprived of water soluble vitamins can immediately be, I would say, witnessed as a deficiency symptoms. One of the most common symptoms, I would say, especially for water soluble vitamins, people would start showing epithelial changes. There will be I would say changes especially in the inner buccal cavity of your mouth. People start having mouth sores. One of the common symptoms for water soluble vitamin deficiency. The moment you start having mouth sore, immediately consider that you are under water soluble vitamin deficiency. So, the moment you start supplementing yourself, so do not postpone it further, you supplement yourself for a little more than a week, the symptoms will recede, okay, you become normal. But for fat soluble vitamins that is not the case. So, it takes longer period to show symptoms and longer time period for you to like really repair, or I would say get yourself rid of those deficiency symptoms. So, that is the fundamental difference. So, quickly look into it. So, they are precursors, whereas they are direct coenzymes, and both are antioxidants, whereas fat soluble vitamins can act more than a coenzyme. In some cases they act like hormones, and fat soluble vitamins are always toxic. So, you could have hypervitaminosis, that is the technical word, when you are on a diet average more than average requirement of fat soluble vitamins. So, you need to like carefully monitor. So, but here since it is non-toxic, they are being eliminated by kidney, and the route of elimination is mostly through renal route. So, since it goes into systemic circulation and absorption across the gastrointestinal tract is also fairly good, and you do have other components that help in the transport of these vitamins across the gastrointestinal tract. Although not mentioned it over here, but it is implied that you require bile salts for the absorption of fat soluble vitamins, whereas you may not require bile as such. You need to have a good source of water soluble, I would say, nutritive diet, and once it goes into systemic circulation and it is made available to the cells and tissues, and later once the job is done, probably it would be metabolized in the liver and eliminated across the renal what do you say structures, and whereas fat soluble vitamins would require bile right from the stage of absorption. Because they are fat soluble, you need to emulsify them, and then they are taken up by the lymphatic lacteals and then goes to the liver where it is stored and then being utilized from there to different parts of our body, and eventually would also find its way to the liver, where it would be metabolized, and then it would be eliminated through gastrointestinal tract. So the unabsorbed material, I would say fat soluble vitamins, would be taken up. And second thing you need to consider, the source of water soluble vitamins. Yes, we are dependent on the gastrointestinal tract microbes, the good ones, which have a symbiotic relationship with us. They are also a tremendous source for water soluble vitamins. So if you could maintain a good gut, you may have a regular supply of water soluble vitamins because they get absorbed and it's available for us. Whereas if our demand for vitamins are higher, then definitely we have to supplement with our diet. So now we are going to take a list of B complex vitamins. Okay, so we have a list of almost 12. I have mentioned a very little number over here. So close to let us say we have maybe 8 or 9, but we do have other vitamins. So as we go across, as we study one by one, we will also look into those minor vitamins. The starting one, we start with thiamine, which is called the B1, then we have riboflavin, then we have niacin, then we have pantothenic acid, then we have pyridoxine, then biotin, folic acid, cyanocobalamin. In addition to that, we also have inositol, then we also have para-aminobenzoic acid. So inositol and para-aminobenzoic acid, although it is not considered as a major B complex vitamin, but it is nevertheless really good one to study because they also contribute in the overall vitamin, water soluble vitamin category. So this discovery is based on the order of discovery. It is basically kind of a chronological order that they have kept. So B vitamins, and one more vitamin we have to consider, the vitamin C, very important one, although I have not mentioned it over here. So we have to consider them as vitamin C. So among these vitamins, you could clearly look at those vitamins which play an essential role, and their deprivation can cause anemia. Okay. So in that way, you have folic acid, riboflavin and cyanocobalamin playing a major role. To be very close, folic acid and cyanocobalamin are, I would say, hematopoietic water soluble vitamins. So any deficiency of folic acid and cyanocobalamin or vitamin B12, immediately we show anemia, symptoms of anemia, although the symptoms of anemia are quite, I would say, specific for each of this vitamin. Generally they go hand in hand. So vitamin B12 will give you like pernicious anemia, okay, and folic acid would give rise to anemia accompanied by neural tube defects, and all of them are required for healthy nerves, okay, and especially vitamin B12 is extremely important for myelin restoration. So in general nowadays the trend would be to supplement vitamin B12 for people who suffer from diabetes at a very early stage, because they are prone for diabetic neuropathy, and probably you can minimize the side effects, although you cannot fully reverse it. And also for elderly population, the geriatric population, it is always recommended that we supplement them with vitamin B12. So more of them when we are going to look up on those vitamins in detail. So to start with, we will pick up on thiamine. Okay, so it is one of the interesting water soluble vitamin to study. So it is also called anti-beriberi factor. Okay, so it is also been given by one more name, what we call it like anti-neurotic vitamin. So it is a very important water soluble vitamin, and if you look at this vitamin is very essential because it modulates variety of metabolic pathways. One of the macronutrients whatever we take, the carbohydrates, the protein, the fat, okay, and other supplementary components like alcohol, which could be endogenous in nature, many of them are dependent on this particular vitamin, thiamine. Okay. So it is required as a coenzyme in enzymatic reaction that involve the transfer of aldehyde group. So this is a nutshell. We are going to see in detail exactly what all the functions, their source and so on shortly. okay, and it's essentially non-toxic. Means what? You won't really run out of any kind of condition called hypervitaminosis if you end up taking more of thiamine. So don't worry if you are under stress, or probably you undergo certain kind of physiological changes, maybe even therapy or diseases, it's okay to supplement yourself with vitamin B1 rather than run out of vitamin B1, because we would require in larger doses, especially during inflammatory process. These water soluble vitamins are very useful. They come very handy in the alleviation of inflammatory responses. So that's a quick glance of what vitamin B1 is all about. Now we are going to look into different aspects, like how specific coenzyme, or what is a specific coenzyme of thiamine, then what are the dietary sources of thiamine, do we have any antimetabolites of thiamine. So when you say antimetabolites of thiamine, that can be monitored in terms of therapy. At the same time, you should also be aware of whether these antimetabolites would have any role to play in terms of interfering with the normal metabolism of thiamine. Okay. So then the daily requirement RDA of thiamine. Then in deficiency of thiamine, what we call it like the classical beriberi symptom. Okay. The most complicated version or elaborated version of beriberi. Then we are going to look at one more important metabolic role that they play, the TPP, thiamine pyrophosphate as we call it. Then what are those people who are being affected, then how do we do investigations, and what is the kind of treatment. Okay, so this would be the glance of different components that we are going to see in the coming sessions, starting from this vitamin B1. So what are the other names? As I have told you, it's also called anurin. So anurin is, you could get the word, you could get the picture, the idea. So since it's having a role with the nerve, the health of nerves, so it's called like deficiency of it, you have a kind of a neurotic conditions, not exactly neurosis, I would say the degenerative nature of nerves. Then thiamine, because thia refers to sulfur, that's the precursor. So you should immediately consider it has some role to play in terms of the thiol group that they possess. Yeah, obviously anti-beriberi factor, and it's a vitamin which contains sulfur in its active form, the thiazole ring is there. So now if you look at the history of thiamine deficiency, the history goes way back to some of the systematic studies carried out by Japanese Navy, okay, during the war period. So Kanihiro Takaki was one of the surgeons in the naval base of Japanese army, and he found especially his crew and seamen who are there on the sea for a longer period of time show symptoms of beriberi fairly quickly. So he came to know like these symptoms are especially prone for people who don't have access to fresh food, or probably the food what they take, or maybe I would say a natural degradation of some of the essential components present in the food. He underwent a systematic study and could make an association that using polished rice, okay, could aggravate, and probably you should take extracts from the rice polishings and could make a therapy out of it. So they were not like really sure what this ingredient was about, but they could make some kind of a potential logical connection between the deficiency symptom and the source of the food that could be given. In other words, how you could give a food as a remedy for the kind of diseases that these seamen and crewmen suffer from. Okay. So all credit goes to Christian Hickman. He was the Dutch physician and he was the professor of physiology. He is the one who actually did a key contribution in, I would say, identifying the exact component involved in the beriberi disease. Okay. So what he did a lot of experiments with fowl chickens. Okay. So he fed them with cooked rice, fed them with variety of rice without, I would say, polishing and with polishing, and he kept doing multiple experiments till he could like practically replicate the symptoms of beriberi in the fowls. Okay. So in fact he was one of the Nobel Prize winners along with other components involved in the overall vitamin research. So, he was one of the key instrumental figure in the early days of vitamin race to identify the key mineral nutrient, or rather I would say the key ingredient that contributes into diseases. So, here you find one more person, Adolf Otto Windus, the German. Okay, so, he is the one who is given the credit of elucidating the structure of thiamine. So, you see over there, it is a very interesting structure. You have a pyramidal nucleus here and you have a thiazole nucleus over here. Okay. So, thio and azole. So, thio refers to the sulfur and azo refers to the nitrogen. So, you see there it is basically a two functional, I would say, compounds, thiazole and a pyrimidine linked together. So, this particular structure has been elucidated by Adolf Otto Windus. Earlier days, nowadays it is more straightforward. You can actually purify the compound, you can do NMR, and if you could crystallize them, you can even collect a fairly good X-ray crystallography picture, and quickly you could elucidate the structure. Way back in the early days, it was extremely challenging because crystallography methods were not fully established and NMR was not completely known, and so people always believed by doing an indirect experiment. It was like a kind of a chemical detection method. So, what they would do, first they will prove the presence of these functional atoms by simple tests, I would say elemental analysis. Then they will proceed further in establishing the functional groups present in them. Then once they are convinced of the functional groups, then they go in part by cracking the structures, okay, of these vitamins. So, they carry out some synthetic reactions and they try to hypothesize the proposed structure and see whether their proposed hypothesis fits into the chemical reaction that they subject the compound to. And finally, as a conclusion, if they have made up the overall structure, they prove it by synthesis. So, a synthetic way of reconstructing the structure would then basically help them to ascertain whatever structure that they have proposed for the compound to be true. And finally, they do bioassays. That is very interesting. So, they do bioassays. So, bioassays give the indication as to what would be the actual structure of that particular compound. So, when they do bioassays, they are sure, they are doubly sure that this compound what they have elucidated would really fit into probably the kind of I would say role that they were proposed to play in a biological system. So, probably this structure after being synthesized would have been subjected to the chicken experiments to prove that this synthesized one has function similar to that of the one which is isolated from a natural source. Okay, so, now we go ahead and we are going to look into the structure of thiamine. So, this is the chemistry of thiamine. So, thiamine consists of a pyrimidine ring. Okay, so, you have a dimethyl, here you have a methyl group and here you have a methyl group. So, here you have a amino group. So, that is why we call it like dimethyl amino pyrimidine. So, this is one structure and this is connected to a thiazole ring. Okay, so, here you have a methyl group and a hydroxyethyl group. So, that is why you end this as a thiazole. So, you say methyl hydroxyethyl thiazole. Okay, they are linked by means of a methylene bridge. So, one of the methyl groups in the dimethyl aminopyridine provides the linking with the thiazole. So, basically you have two heterocyclic nucleus, the first nucleus being a pyrimidine nucleus and the second one being a thiazole nucleus. Okay. So, these two independent nucleus are being linked by a methylene bridge linkage. So, if you look at what is the role of thiamine. So, thiamine needs to be loaded, something like when you have a gun in your hand, you take the bullet and you put it up, and before you load the bullet you remove the wrapper and keep it in such a way that it would be ready for firing. So, thiamine becomes the actual thiamine, the functional thiamine, because thiamine is basically the precursor and TPP is actually the functional version of thiamine. So, what is this TPP? So, it will use one of the key contributors of phosphate group, obviously the ATP molecule, adenosine triphosphate, and this would provide, I would say, 2 phosphate groups. So, in other words, thiamine would be phosphorylated. Okay, so that is what we call it, like thiamine pyrophosphate TPP. 2 phosphate groups are added. So, you see over there, here is one phosphate group and here is one phosphate group. So, a dual phosphate group to any organic molecule is called a pyrophosphate. Okay. So, here you have a substituted pyrimidine derivative, here you have a substituted, I would say, thiazole derivative, and then to this you are adding 2 phosphate groups, and these phosphate groups, since they are dual in nature, they are called pyrophosphates. So, together you call this entire structure as thiamine pyrophosphate. Okay. Now, this particular reaction is being catalyzed by a transferase enzyme. So, that is why we call it like TPP transferase. TPP transferase uses ATP as a source for phosphate and transfers this to thiamine pyrophosphate, resulting in AMP, okay, which is a second messenger, which in turn can also be used as a second messenger signaling component along with the other cytoplasmic components present inside the cell. Now, we are going to look at various sources of thiamine. Okay. So, as I have been telling you, one of the richest sources for B vitamins, especially water soluble vitamins, would be food grain coatings, rice, wheat, yeast. That is one of the reason in the modern day we try to improve the quality of rice, as we say it, as we call it, so called way of improvement of rice is basically removing all the polishing. So, the reason why we polish rice is that we want to make the carbohydrate available for energy. In other words, on our goal of making the macronutrient available readily. Okay. So, whereas if you cook a polished rice, it cooks faster than if you cook an unpolished rice, but that comes with a price. Okay. So, unpolished rice would delay in cooking, may not be a quick snack. So, for that reason we go for polishing, but once you polish you also remove all these essential vitamins like thiamine from the surface. Okay. In addition to that, what we call it, like you can have any old cereal with all the coatings in it and all the pulses with all the coatings in it would be an excellent source for thiamine. Okay. In addition to that, nuts, yeah, and oil seeds as well. So, they are also, especially the outer coating, they are all rich in I would say thiamine. So, you do have thiamine also from, I would say, animal sources like meat, liver, egg and fish, you do have thiamine, but vegetable source, especially the cereals and grains, is always the good source for water soluble vitamins. So, as I have been telling you earlier, whenever you make a food preparation, and they do, especially the commercial preparations, if they find it is going to get deprived of certain nutritive material, we fortify them with essential components. So, it is one of the common practices in the market to fortify the food material with water soluble vitamins. So, that would be basically a good way of enriching the food. Now, if you look at in detail, like what all different ways by which this thiamine is being made, we do not make thiamine. Actually, we depend solely on the dietary source of thiamine. So, you see there, thiamine is basically being synthesized by plant and by microorganisms, and yeah, very few, I would say, animals, I would say mammals or rodents are capable of producing thiamine. In general, human beings do not produce thiamine at least to the level required. In fact, generally it is being accepted that human beings do not actually produce or synthesize thiamine to an appreciable level. Probably it could be a very little recycled version of thiamine available from what do you say, metabolic pathway. We do not exclusively produce thiamine, but this thiamine is actually produced in sufficient quantity from the bacteria present in the gastrointestinal tract, okay, the gut bacteria as we call it. Okay, as I have been telling you, yeah, the wheat flour unpolished rice are all good sources, and we have already seen this. And you see, the body can store a maximum of 30 milligrams of thiamine at any given time in its tissues. Okay. So, that is one of the reason, being water soluble, and our body, I would say, as you see, all the trillions of cells are soaked with water, then it has been like kind of recirculated. So, our body would be like fully flushed with water every now and then, but at any given time it has been fully soaked with water. So, the vitamin reserves, especially thiamine reserves in the cells, can be washed away through a normal filtration process across kidneys. Okay. So, as you see over there, so these are the major sources, the rich sources, the good ones, and the fair sources. So, if we have to have a diet rich in, I would say, a balanced diet rich in vegetables and grains, then we adequately get a dietary source of thiamine for a longer period of time. Now, as you look at the thiamine occurrence, as I have been telling you, where you find large quantity of thiamine, mostly it is present in the skeletal muscles, heart, liver, kidney, and brain. Now, before we conclude today's session, I want to give you a word. So, when you take the food and process it, you are likely to destroy the vitamin at a very high temperature. So, take care that you do not overcook your food and lose all thiamine sources. So, in the coming sessions we will go into more detailed study of thiamine. Thank you for your attention.